When you think about sugar, you probably just picture those little white crystals, right? The stuff you put in your coffee. But, at the molecular level, these things are incredible shapeshifters. They're constantly twisting and transforming in this secret chemical life. And today, we're going to pull back the curtain on all of it.
All right, so here's our game plan. We're going to start with the basics, like what a carbohydrate even is. Then we'll meet its simplest form. From there, it gets really cool. We'll dive into the wild 3D shapes these molecules can make, watch them do a magic trick in water, and finally, see how they get modified to do all these amazing specialized jobs in nature.
So first things first, what's in a name? The word carbohydrate sounds kind of like hydrated carbon, which is a little bit of an old school misleading term. So let's get into what it really means from a chemical point of view.
Okay, so here's the real definition. A carbohydrate has a backbone of carbon atoms, and hanging off that backbone are a bunch of alcohol groups, what chemists call hydroxyl groups. But the real key, the thing that makes it a sugar, is that it's capped off by one of two special groups, either an aldehyde or a ketone. That specific structure is the secret to everything these molecules can do. Now let's meet the simplest members of this family, the monosaccharides. You can think of these as the fundamental Lego bricks of the sugar world. They're the single units that can't be broken down any smaller. The way we classify these is actually super straightforward. To figure out what kind of monosaccharide you're looking at, you just have to ask two simple questions. One, is its special group an aldehyde or a ketone? And two, how many carbons does it have in its backbone? That's it.
And here's that first big difference in action. An aldose, like glyceraldehyde here, has that important reactive group right at the very end of the carbon chain. But a ketose, like dihydroxyacetone, has that same kind of group tucked away somewhere in the middle. Just that one little difference in placement is what defines the two major families of simple sugars. But this is where things get really, really interesting. We have to stop thinking about these as flat drawings and start seeing them in 3D. Because with sugars, you can have the exact same set of atoms arranged in different ways in space, creating this whole universe of molecular doppelgangers we call isomers. So this is the big idea, stereoisomers. It all happens because of something called a chiral center. That's a carbon atom that's attached to four completely different things. And because it is, you can arrange those four things in two different ways that are mirror images of each other, creating two totally distinct molecules, even though they have the exact same chemical formula. This is the perfect example. Look at D-glucose and L-glucose. They are perfect mirror images. It's just like your left and right hands. They look the same, but you can't perfectly stack one on top of the other. And this might seem like a tiny detail, but in biology, it's a huge deal. Our bodies are built almost exclusively to recognize and use the D forms of sugars. So just how many different versions are we talking about here? Well, for a common sugar like glucose, which has four of these special chiral centers, the math just gets wild. It leads to a staggering 16 possible stereoisomers. I mean, think about that for a second. 16 completely unique molecules, all made from the exact same parts. And out of those 16, here are just half of them. These are the 8D forms, the ones that are most common in nature. From allose all the way to talose. Every single one is a unique sugar with its own properties. And the only difference is that subtle left-right arrangement of those little OH groups. It's just an incredible example of molecular diversity. Okay, so prepare yourself for a major plot twist. All those straight chain-like drawings we've been looking at, well, for the most part, they're kind of a convenient lie. The second you put sugar in water, something really amazing happens. Yeah, you heard that right. When it's in a solution, like the water inside a living cell, less than 1% of glucose molecules are actually floating around in that straight chain form we've been drawing. The vast majority, almost all of them, take on a totally different shape. And that fact changes absolutely everything about how sugars work. This is the key right here. It's a dynamic equilibrium. The sugar molecule isn't just sitting there. It's constantly flipping back and forth from that unstable open chain form to a much, much more stable closed ring. It's like this constant chemical dance between the two shapes. So how does this transformation actually happen? Well, the molecule literally bites its own tail. You can see it here. An alcohol group from way down the chain is attracted to the aldehyde group at the very top. It reaches around, forms a new bond, and snap! The chain closes up into a stable ring.
And this act of forming a ring, it creates yet another layer of complexity. That carbon atom that used to be the aldehyde, it now becomes a brand new chiral center, and it gets a special name, the anomeric carbon. And since it's a new chiral center, it means the ring can snap shut in two slightly different ways, creating a whole new pair of isomers called anomers. So let's meet them, alpha and beta glucose. They look almost identical, right? But look really closely at that anomeric carbon. That's carbon number 1 on the far right. In the alpha form, the new hydroxyl group points down. In the beta form, it points up. And believe it or not, that one tiny detail, up versus down, determines whether sugars link together to build something soft like starch or something super strong like cellulose.
But wait, nature isn't done yet. These basic sugar rings, they're really just the starting point. They can be chemically modified, almost like they're being decorated with new functional groups to create a whole new cast of characters with super specialized jobs. Think of it like a molecular toolkit. You can tweak a group to create a sugar acid, or you can make a sugar alcohol. Or, and this is a really cool one, you can swap out a hydroxyl group for an amino group, creating an amino sugar. Every single one of these modifications creates a new molecule, a new tool for a new biological job. And here is just a fantastic example of how powerful this is. You take glucose, you swap just one of its hydroxyl groups for an amino group, and you get something called glucosamine. Then, when you link a bunch of those units together, you build chitin, the incredibly tough, resilient material that makes up the exoskeletons of bugs and shrimp. So when you step back and look at the big picture, it's just amazing. From a simple carbon backbone, this entire universe of complexity just emerges. Through all these different tricks, 3D arrangements, ring formation, functional tweaks, sugars generate this huge vocabulary of shapes. And those shapes are the basis for everything, from storing energy to building the structure of trees to even letting our cells talk to each other. It all starts with one simple shape-shifting molecule.